We continue our recorded session on meiotic cycle, meiotic division, by looking at the events that are specific to gametogenesis in males and females. No? So, as mentioned, meiosis is the division of the nuclear material, the nucleus, um, um, specific to sex cells, to gametes, which will produce ova or egg in females and spermatozoa in males. Okay. The key difference I've already discussed in part one of this recorded session is the reduction of the chromosomal complement of gametes from the diploid state to the haploid condition. So that's a key difference of meiosis when compared to mitotic cycle. So during meiosis 1, synthesis of DNA will precede the actual chromosomal events, thus crossing over, segregation, no? And starting with the diploid set of chromosomes, after homologous chromosomes do the crossing over, they will separate and will be distributed in each daughter cell with a halved number or half number of the original chromosomal set. Okay, so the chromosomal complement of the daughter cells we expect will be 1N to C. No? Unlike starting with 2N 4C chromosomal complement. During meiosis 2, the processes are similar to mitotic division. Each of the previous two cells will produce genetically identical daughter cells that can now be properly called gametes, the ova or the spermatozoa. The chromosomal complement of the daughter cells after meiosis 2 will be transitioned to or will be converted to 1N 1C from 1N to C. Okay, so this is just an illustration of the next two slides are just illustrations of meiosis 1 and meiosis 2. Okay. Right, so now let's take a look at the processes of spermatogenesis and oogenesis. Spermatogenesis takes place in the the testicles of um, the male animal, okay, and particularly in the structures called seminiferous tubules, which are coiled tubules within the testis. Here you can see the right. So here, this one that I'm highlighting are seminiferous tubules. If you cut across a tubule, it would look like this. So you can see along the the order or the direction of maturation is from the lining, outer lining of the tubule going towards the lumen. So the mature sperm are found in the lumen and are expected to congregate there um, until the time comes that they are ready for transportation um, out of the animal body, out of the male animal body. Okay, so let's take a look at the stages. Right, so we have specific names or specific types of cells for all the stages. So during mitosis, your spermatogonium, spermatogonia, which is in diploid state, right, will is expected to possess a diploid set of chromosome, and after meiosis one, or as the spermatogonium matures, okay, it will transition to become a primary spermatocyte, and that primary spermatocyte will carry out or undergo meiosis 1. So from being a primary spermatocyte, it will now become a secondary spermatocyte, which is already haploid and haploid in number, no, in terms of the chromosomal complement. Meiosis 2 will produce early spermatids, these four here, one, two, three, four, which are already the the male gametes that are already haploid in state, and during a process called spermiogenesis, these early spermatids will continue to mature, will become late spermatids, and our sperm ready immature and then later on mature sperms that are ready for transportation, ready for union with an egg cell to produce a zygote. Okay. Um, okay, so here, so just remember the cell types per stage associated to specific stages of the meiotic cell cycle. Okay? Again, just a rundown. So your diploid spermatogonium will transition to becoming a primary spermatocyte. Your primary spermatocyte will divide to produce secondary spermatocytes, which are haploid in number. And these secondary spermatocytes will undergo meiosis 2 to produce spermatids that later on will mature as spermatozoa. Okay. Now, for the case of female animals, the process of egg development is called oogenesis, and it takes place in the ovaries. Okay. Similar with spermatogenesis, so we have specific names or specific cell types per stages associated to the meiotic processes, right? So, um, one thing that I'd like to emphasize is that unlike unlike the male animal, female animals will have a finite number of their oogonium or oogonia, the stem cells that are responsible for the production of the egg. There is a finite number of these egg cells, and they are produced, or the number is determined even before the female animal is actually born. So there is a specific number of oogonia that a female animal already has. Okay, even before that female animal is mature. Okay, so the oogonium will have a diploid complement, a diploid set of cells, all right, a diploid state, okay, and during meiosis 1, we expect the having of the chromosomal number into 2, right? And your primary oocyte is uh, the expected um, input cell prior to meiosis, prior to the meiotic cycle 1. This primary oocyte is arrested during prophase 1. Okay, and once that childhood ceases, the female animal is now sexually active, or sexually active in the sense that the female animal will release one mature egg per cycle. Okay, this primary oocyte will continue to mature, will continue to grow, and produce a secondary oocyte, which is actually arrested in meiosis 2, no? And once ovulation takes place, this secondary oocyte will be released, and if a sperm will be able to fertilize it, it will complete meiosis 2. Okay, so the completed or the reconstituted um, cell, or the fertilized egg, okay, will produce an ovum that is haploid in number in terms of the chromosomal complement. Okay, and will continue to mature, will continue to divide as the animal progresses or the zygote keeps on developing. Okay. Now, unlike spermatogenesis, in oogenesis, the other three gametes, okay, are actually non-functional. So these other three gametes here that are produced, the first polar body, the second polar body, and the other degenerate polar bodies, they are non-functional. They carry the haploid number, the other haploid number of chromosomes, but they are not functional. Okay, so please take note of this key difference between spermatogenesis and oogenesis. So the polar bodies are not really as functional as complete and not as functional and as complete when compared to the completed or to the fertilized secondary oocyte, no, that becomes the ootid. Okay. Okay, so I mentioned this. Okay, so just a rundown again. So your oogonium, okay, uh, you have a finite number of these even before you're born, right? Uh, the oogonium transitions to become a primary oocyte, which is diploid in number, right? Then during puberty, secondary oocyte will be released um, in relation or after the meiotic cycle 1. Okay, that secondary oocyte has a complement of another of one polar, the first polar body. If secondary oocyte travels through the female reproductive tract and then encounters a sperm, okay, it will be fertilized. The chromosomal complement will be reconstituted to diploid, okay, and that ovum, right, um, uh, after meiotic cycle 2 will of course produce the other polar bodies, other degenerate polar bodies, and the second polar body. Okay, again, this is a juxtapositioning of another rundown of the stages of egg cell development in female animals. Okay, so as a review, this table summarizes the key differences between mitotic and meiotic cycle. So in terms of the type of cell, meiosis happens in gametes. In terms of the number of divisions per cycle, there are two divisions in meiosis compared to mitosis. In terms of the number of daughter cells that are formed, we expect meiotic cycle to give rise to 4 progeny cells, unlike in mitosis, which only gives rise to 2. In terms of the chromosomal complement, meiotic cycle will produce progeny cells that are haploid in state. Right, and during time of cycle, or in terms of the time of cycle, mitosis, which happens in somatic cells, will continue on happening throughout the life or throughout the lifespan of the animal, while for meiosis usually completed after sexual maturity, especially in females. Okay, and unlike mitosis, meiosis, of course, is important for sexual reproduction and the generation of new gene combinations or the increase in the diversity of the genetic pool within the animal species. Okay, so that ends our recorded session on meiotic cycle.